Biogad, simply more knowledge. A podcast not just for biogeeks. Informative and interdisciplinary.
In today's episode, we're diving into the utilization of energy. How did it even come about that plants conduct photosynthesis? Who actually invented photosynthesis? And what conditions were necessary for it to evolve? How do cells harness energy? Do they all do it the same way? In this episode, and the next, we'll be exploring answers to all those questions. But today, we're not covering the origin of the first Archaeon. That topic remains full of mysteries. Still, to make such an origin about 3.5 billion years ago possible, certain preconditions had to be in place. One of those was chemical evolution during the Hadean Eon. The Hadean is the time span on Earth from its formation until about 4 billion years ago, a period lasting roughly 600 million years, extremely distant in the past. During the Hadean, life did not yet exist. This eon marks the earliest portion of what we call the Precambrian, the time before the Cambrian. Our understanding of chemical evolution largely rests on various hypotheses. One key hypothesis is abiotic formation of biomolecules, that complex molecules essential for life emerged from inorganic precursors. How exactly that happened remains unresolved. Yet we can envision a plausible multi-step pathway. First, simple organic molecules, alcohols or acids, likely formed. By definition, organic molecules contain carbon, oxygen and hydrogen. Once simple organics existed, the building blocks of biomolecules could arise. Monosaccharides, simple sugars, amino acids, and nucleotides. Important note, don't confuse nucleotides with nucleoids. A nucleoid is a structure in prokaryotes similar to a nucleus containing freely floating DNA, whereas nucleotides are the chemical units in DNA and RNA, consisting of a sugar, an organic base, and a phosphate group. With these basic building blocks in hand, more complex organic molecules could emerge: dye and polysaccharides, proteins, nucleic acids, providing essential prerequisites for later living cells. How exactly these biomolecules assembled into cells is still uncertain. Some researchers suggest mineral pores in rocks formed primitive cell-like compartments. But to keep things moving, we'll skip ahead and see what happened once the first cells appeared. Now we enter the Archaean Eon, the middle Precambrian. Here, biological evolution began with prokaryotes. Without prokaryotes, there would be no eukaryotes. But how could eukaryotes ever arise? How do we know prokaryotes were such ingenious innovators? In this episode, I'll walk you through the evolutionary changes that enabled the astonishing diversity of life we see today. And those changes center on energy usage. Prokaryotes revolutionized it. The first cells very likely originated in what we call the ocean, about 3.5 to 4 billion years ago. Since then, unimaginable transformations have occurred. Yet one thing has hardly changed. the bioenergetic processes cells use to obtain energy. What we observe everywhere are metabolic pathways, both aerobic, with oxygen, and anaerobic, without oxygen. We can approximately trace the evolution of bioenergetics by studying extant chemolithotrophic prokaryotes and analyzing Precambrian geologic sediments. Chemolithotrophs can't get energy from organic molecules. They must tap into inorganic sources. Prokaryotes paved the way for eukaryotes. The final segment of the Precambrian is the Proterozoic, during which eukaryotic cells, those with true nuclei, evolved sufficiently to give rise to increasingly complex organisms. Fossils from the Precambrian are rare, but include stromatolites in Western Australia, dating back 3.5 billion years, formed by cyanobacteria, from organic origin, not inorganic. Another is the Ediacaran fauna near the end of the Precambrian. These fossils document multicellular life existed around 570 million years ago. 30 million years later, approximately 540 million years ago, the Cambrian began, ushering in the Cambrian explosion when nearly all animal phyla emerged. To understand bioenergetic evolution, we need to study geological layers laid down before the Cambrian. That's tricky because most have been altered by tectonic forces. Back then, the chemical environment and climate were radically different, sometimes resembling modern deep-sea black or white smoke events. Life on Earth may also have been influenced by asteroid or comet impacts, though evidence remains inconclusive. So now it's time we take a closer look at the chemistry. To follow the upcoming explanations more easily, we'll need a bit of background knowledge about reduction-oxidation reactions, also called redox reactions. In these reactions, substance A reacts with substance B. One of these substances donates electrons and the other accepts them. These two processes, donation and acceptance, always occur together. I'll try to keep it brief, but we do need to understand a little more chemistry. So, what does oxidation mean in chemistry? When a particle is oxidized, it gives away electrons. A reduction is the opposite. The particle gains electrons. So oxidation is the loss of electrons, and reduction is the gain of electrons. But what does all this have to do with energy? When electrons are released, energy is also released. And how can we tell? Well, usually the environment where the oxidation takes place gets warmer. That's how we notice energy being released. Now, it's important not to confuse the terminology. The particle that donates electrons is called the reducing agent, or the donor. The particle that accepts the electrons is the oxidizing agent, or the acceptor. This oxidizing agent accepts the electrons and oxidizes the reaction partner by taking its electrons away, basically saying, hand over those electrons. Now, there's something else we need to understand about how energy is utilized. Oxygen doesn't have to be involved in an oxidation. Since it's all about electrons, the presence of oxygen isn't a requirement. That might seem a little misleading because the term oxidation sounds like it must involve oxygen. After all, the scientific name for oxygen is oxygenium, but again, no oxygen needed. Also, hydrogen doesn't have to be released during these reactions either. Hydrogen is written as a capital H for hydrogenium with a superscript plus sign, H superscript plus. This H plus is also called a proton. So, just to clarify, the terms hydrogen atom, hydrogen ion, hydrogenium, H plus and proton all basically mean the same thing in this context. You can treat them as synonyms here. If an H plus ion is released in a reaction, it usually doesn't stay alone for long. It quickly binds to another particle. It might even bind with another H plus. And what does H plus give us? It gives us hydrogen 2. That's a capital H with a subscript 2. Important side note, it really does matter whether a symbol is written as a superscript or subscript. H2 is molecular or free hydrogen. As soon as two atoms are joined together, we call it a molecule. Because hydrogen 2 is so reactive, it's often involved in oxidation reactions. It acts as a reducing agent. Now, hydrogen can be released through various reactions, but there are also other reducing agents besides H2. For example, there are carbon-containing compounds like lactate and acetate, or alkali and alkaline earth metals like potassium, K, sodium, Na, magnesium, Mg, zinc, Zn, and so on. Typical oxidizing agents include hydrogen peroxide, ozone, chlorine, iodine, and others. We humans use them as disinfectants and bleaching agents. Hydrogen peroxide will come up again in a later episode when we talk about homeostasis and the liver. Let me briefly explain one example of a redox reaction. Oxygen has a very high oxidation potential. That means oxygen really likes to accept electrons. So, oxygen is an oxidizing agent. It can, for instance, oxidize iron. And what happens when iron gets oxidized? It starts to rust. The sand in the Namib Desert is so red because it contains a lot of rusty iron oxide. In that reaction, iron gives up its electrons and therefore acts as the reducing agent. Oxygen is reduced, iron is oxidized. This high oxidation potential of oxygen is one of the reasons why the way cells gain energy had to evolve over time. What evolutionary stages can we reconstruct today? I've grouped them into 13 stages, starting with stage 0, the starting conditions about 4 billion years ago. Then stage 1, stage 2, and so on. So here we are at stage 0, the initial state roughly 4 billion years ago. The environment at that time, the primordial ocean, and within this primordial ocean, there were inorganic particles and, this is crucial, a wealth of energy-rich, hydrogen-rich organic molecules. These molecules had formed through geochemical processes. So, we're in the Hadean Eon. The formation of rocks was one of the key preconditions for life to emerge. I won't go into how rocks formed here, that would take us too far afield. But thanks to rock formation, the primordial ocean effectively turned into a kind of nutrient broth. And suspended in this broth were the biomolecules I mentioned earlier. But what wasn't present in the early atmosphere, and likely not, or only barely, in the primordial ocean, was free oxygen, O2. This strong oxidizing agent simply wasn't around in significant quantities. Now, since 2024, we have evidence suggesting that in certain marine regions, like the Clarion-Clipperton zone in the Central Pacific, manganese nodules may have split water, H2O, into its components via electrolysis. This process would have generated free oxygen in the deep sea, sometimes referred to as dark oxygen. But manganese nodules aren't found across the entire ocean floor, so this process was limited in scope. That means prokaryotes had to find another solution to the oxygen shortage. And that takes us to the next evolutionary step. Stage 1. We're now in the Archaean Eon. In the primordial ocean, organic molecules such as glucose, C6H12O6, are floating around. That's 6 carbon atoms, 12 protons, and 6 oxygen atoms. And among these glucose molecules, single-celled organisms, prokaryotes, are swimming about. They used glucose as an energy source. We humans like glucose too, especially when we're trying to focus during an exam and need a quick energy boost, right? The process of breaking down glucose to extract energy is called glycolysis. During glycolysis, a phosphate group is added to an organic molecule. This process produces ATP, adenosine triphosphate. Importantly, glucose is broken down without the need for oxygen. Glycolysis is an anaerobic process, and in the process, free protons, H+, are released. These hydrogen ions began to accumulate in the primordial ocean, but protons don't like to stay on their own. They want to bind with something. And therein lay the problem. There was little or no free oxygen around. If O2 had been present, it could have reacted with H+ to form water, H2O. But without oxygen, that wasn't possible. Now we arrive at stage 2, the solution to this problem. Without O2 in the ocean and with an excess of protons H+, hydrogen had to be incorporated into other molecules, specifically organic molecules. This led to the formation of organic acids, such as lactate and acetate. These acids began to accumulate, not only in the ocean, but also inside the prokaryotic cells themselves. This led to a new problem. The acids caused the internal pH of the cells to drop. In other words, the cells became more acidic, a process known as acidification. Let's pause here to clarify. What is pH? It's the negative base 10 logarithm of the hydrogen ion concentration in a solution. So, the lower the pH value, the more H+ ions are present. The pH value is a measure of how acidic a liquid is. The P in pH stands for potentia, Latin for power or potential, and the capital H stands for hydrogenium. If you wrote both letters as capitals, pH, you'd actually be writing the abbreviation for physics class in Austria. Now, acidification can also happen in humans. If our blood becomes too acidic, it leads to a dangerous condition called acidosis. That can result from liver or kidney failure, tumors, or anaerobic glycolysis, the very process we've been discussing. So what was the solution for prokaryotes? This brings us to stage 3. Acidification wasn't any more fun for prokaryotes than it is for us. They had to get rid of the excess protons somehow, and they managed to do that by evolving a pump embedded in their cell membrane. This pump was powered by ATP. It doesn't run on its own, it needs energy. And that energy came from ATP, which was already being produced during glycolysis. This proton pump actively transported surplus H+ ions out of the cell, allowing prokaryotes to prevent internal acidification. But why is acidification such a big problem anyway? Because metabolic processes only function within a specific pH range. There's very little wiggle room. Even slight shifts in pH can cause enzymes to fail. However, this proton pumping solution created a new challenge. It was so successful that the prokaryotes began to multiply rapidly. And that caused a resource problem. They were consuming organic molecules faster than geochemical processes could replenish them. As competition increased, ATP became precious. The days of freely burning ATP were over. We now reach stage 4. At this point, a major driver of evolution came into play: natural selection. When two organisms compete for the same resource, the one who can make more efficient use of it, or who finds an alternative route, has the edge. In this case, the goal was to get rid of protons. Some prokaryotes figured out how to do that without using up ATP, and that gave them a clear advantage. What does such an advantage mean in evolutionary terms? A higher reproduction rate. These prokaryotes could multiply more quickly than those that still needed ATP to get rid of protons. Of course, these organisms didn't sit around discussing and inventing strategies. Instead, mutations must have occurred that enabled this capability. Over time, these mutations led to the evolution of different electron transport systems in prokaryotes. Some developed simple systems within their cell membranes that relied on just two molecules with differing redox potentials. And just like that, prokaryotes had succeeded in creating electron transport chains without using up ATP. With that foundation, they could now start to refine and improve these systems.
If you enjoyed this episode, please give me a thumbs up. If you have any constructive feedback for me, feel free to send it to biogad@outlook.de. Thank you.
This was an episode of Biogad by Ulrike Randl-Gadora, covering various topics in biology.
The text was originally written in German and then translated and narrated by AI. If you notice any translation errors, please send an email. Thank you.